We are very fortunate in that we have Dr. Oliver Witard joining us from King's College London. Dr. Oliver Witard is a senior lecturer in exercise metabolism and nutrition, and he's going to be taking the opportunity to speak to you guys today about protein from animal and plant-based proteins and what's the difference for muscle growth. Ollie, we're really pleased to have you with us. We're really excited for the talk today. I can see my camera's given up, but that's because we're trying to ask it to do too many things at once. So I'll just politely switch myself off. And yeah, basically, mate, over to you. And thanks very much for joining us today.
So this presentation, as Mike mentioned, is called Protein from Animal and Plant-Based Proteins. What's the difference for muscle growth? And I think that this is a relatively topical question, given the fairly recent documentary that received a lot of attention, at least in the field of sports nutrition, which was the Game Changers documentary, which I think, in summary, had a number of important points that it was trying to make, but unfortunately it decided to go perhaps and become go over the top almost, and sensationalize some of the main messages rather than sticking to the evidence base, the scientific evidence base. So really what I'd like to do in this presentation is to present the science behind this question in terms of comparing plant versus animal proteins in terms of stimulating muscle protein synthesis, which we know is a key aspect of, from a metabolic basis, for controlling muscle growth. But before we get into the specific question, and I appreciate that this is very much revision for this audience, but I think it's always worth highlighting, reminding ourselves of the importance of skeletal muscle mass and quality, and with the view of really highlighting that muscle mass and quality, the importance of which extends beyond athlete performance to also health and well-being. So on the top of this slide, we see a number of different areas of context within performance nutrition, where of course muscle mass and quality is important either from a performance perspective or an aesthetic perspective. So of course, if we take the centre back in football as just one example, we can take the sprinter with a high power to mass ratio, we could take the judoka player, high quality weight loss, maintaining that muscle but losing fat mass scenario. We may take ballet dancers for, of course, aesthetic reasons, that muscle's important. We may take bodybuilders as well, again, for aesthetic reasons, but then also more occupational scenarios such as combat, military combat, for example. So we've got a whole host of different scenarios in which skeletal muscle mass and quality, of course, is important. But beyond performance, and I say that loosely because it depends how you define performance, but we also have a role of skeletal muscle in terms of health. Healthy aging, of course, receives a lot of attention. As we age, we get a loss of skeletal muscle mass. So if we can maintain that muscle with advancing age, it's possible that we can live longer or at least healthier lives. Of course, muscle's important in terms of child growth, in terms of healthy weight loss in more of a weight management scenario, and also in terms of other cardiometabolic health outcomes, such as reducing the risk of diabetes. We know that skeletal muscle tissue is a key tissue in terms of taking up glucose from the bloodstream. And all of this kind of context is really nicely summarized in a paper from Bob Wolf back in 2006, published in the American Journal of Clinical Nutrition, which was titled The Underappreciated Role of Muscle in Health and Disease. So if we talk about muscle mass, also worth noting before we get into the specific question is at the metabolic level, the main player in terms of regulating skeletal muscle mass, at least in otherwise healthy individuals, is the stimulation of what we call muscle protein synthesis. Now, muscle protein synthesis refers to the synthesis of amino acids, either from the blood or the intracellular pool of the skeletal muscle itself, into bound functional muscle proteins. And these muscle proteins can be crudely classified either as myofibrillar proteins, so your actin, myosin, tropomyosin, that are responsible for changing muscle mass, essentially, or your mitochondrial proteins. So these are the energy producing proteins. Plus we have collagen proteins as well, so more structural proteins. In addition to muscle protein synthesis, we have the converse metabolic process, namely muscle protein breakdown, which refers to the breakdown of bound muscle proteins back into their amino acid precursors. And essentially it's the balance between muscle protein synthesis and muscle protein breakdown which determines whether we gain muscle mass or we lose muscle mass. But in otherwise healthy individuals, primarily it's this right-hand arm, muscle protein synthesis, i.e. if we can increase the response of muscle protein synthesis to an exercise or nutritional intervention, is primarily responsible for the change in muscle mass. Now, this is a slightly different story when it comes to more compromised populations, for example, cancer cachexia patients, sepsis, burns patients, etc., where muscle protein breakdown plays a much larger role. But in this context, with athletes, then changing this metabolic process or regulating muscle protein synthesis is seen as the primary driver of a change in muscle mass. But before we can start understanding how we can optimize this response of muscle protein synthesis with, for example, protein feeding, it's important to understand how the magnitude of muscle protein synthesis is regulated on several different levels of physiology. So what this slide does is, at the top, it has an ingested protein source, and at the bottom, it has muscle protein synthesis rates. And essentially, we're going to work from the top down on this schematic, and we're going to fill in the gaps. So what we're doing here is determining the key factors from a physiological perspective, which determine how well this ingested protein source is able to stimulate muscle protein synthesis rates. And the first factor is determined by blood amino acid availability. So essentially, blood amino acid availability is dictated by three subtopics, namely the digestibility of the protein source, the amino acid absorption kinetics of that protein source, and also the splanchnic retention of those amino acids. In other words, what percentage of the amino acids derived from the protein are retained in the gut rather than being transported and appearing into the bloodstream. And we're going to come back to each of these factors when we look at different types of proteins or different sources of proteins. So hold this slide in your minds when we come to looking at mechanisms of action. The second key physiological factor, now that we've got the amino acids from the protein in the bloodstream, relates to the delivery of these amino acids to the skeletal muscle itself. And this process, this metabolic process, is essentially under the control of insulin and is determined by, again, three subtopics, that of blood flow, capillary recruitment, and also microvascular perfusion. And of course, exercise is a key mechanism that we can change blood flow and also change microvascular perfusion. Is relating to taking up these amino acids into the muscle itself. And what's key here are transporters, much like other aspects of metabolism, whether it be carbohydrate or glucose metabolism, with GLUT4 being a key transporter. The same is true within protein metabolism and amino acid uptake into the cell. And one of the key amino acid transporters is called LAT1. So LAT1 is on the membrane of the muscle and is the transporter for leucine, but also other amino acids as well. And then the fourth level of physiology is anabolic cell signaling, which essentially refers to almost like a traffic lights type of scenario. So inside the muscle, we have these intracellular muscle proteins, one of which is called mTORC1, which stands for the mechanistic target of rapamycin complex 1. We've got proteins upstream of mTORC, and we've got proteins downstream of mTORC. And essentially these intracellular muscle proteins work as a traffic light signal to say whether we should switch on this process of muscle protein synthesis or whether we should downregulate this process. So essentially this slide quite, I hope at least, provides a nice overview of the physiology that underpins how well an ingested protein source is likely to stimulate muscle protein synthesis rates. So there's the introduction. So what we are going to do in this presentation, in the main body of the presentation, really is trying to address 3 key points. First of all, we're going to establish the key factors that determine the anabolic potential of a protein source. So essentially revisit some of the introduction. We're then going to look at the scientific studies that have directly compared the anabolic capacity, so essentially the measurement of muscle protein synthesis, between animal-based protein sources and plant-based protein sources, whether they're consumed in food form or in supplement form. And then finally, we're going to talk about this concept of protein quality and essentially where next for scientific research in this field, given that I know there are several PhD students, postdocs that are in the audience. So first of all, let's establish the key factors that determine the anabolic potential of a protein source. And essentially there are 3 key factors that determine the potential of a protein source to stimulate muscle protein synthesis during exercise recovery. And they are, first of all, in the red, you've got the digestion and absorption kinetics. So the digestion refers to the percentage of amino acids that are derived from the protein that can essentially make it into the circulation. Whereas absorption kinetics refers to the speed at which those amino acids can make it into the bloodstream and also the time course of events. The second factor relates to essential amino acids in the context of the profile of the essential amino acids of the protein source. So whether they've got a full all 9 essential amino acids, and also the content. So essentially the abundance of those essential amino acids or the percentage of that protein source that contains essential amino acids. And then the third factor relates to the leucine content of the protein source. So we're going to, in the next few slides, going to walk through why each of these factors are so important, presenting some of the mechanistic studies that have been published in the field of muscle protein metabolism. And I have selected these based on, due to time restrictions, of course, can't present all of these mechanistic studies. So first of all, let's look at the digestibility of various commonly consumed protein sources. And essentially for this figure and for many of the figures that are following up, on the left-hand side, we've got the animal-based proteins. And on the right-hand side, we've got the plant-based proteins. So the plant in green, and then the animal proteins either in red or in white. And essentially the dairy proteins are those that are in white. And as you can see, on the y-axis, we've got protein digestibility expressed as a percentage of the amino acids within that protein source that make it into the circulation. And without exception, you can see that the animal-based protein sources are essentially more digestible compared to the plant-based protein sources. And of course, this is explained by the anti-nutritional components of many plant-based protein sources. What we see in this slide is the differences in the amino acid kinetics between different protein sources. I've selected just one example here, and essentially I'm comparing 2 different dairy proteins, namely whey protein, which is a fast protein, which is soluble and readily available into the blood, whereas compared to casein, which actually clots in the stomach and actually has a much slower absorption kinetics. But what I'm presenting here is a study by Dan West and colleagues during his time in Stuart Phillips' lab in McMaster, Canada, which was a really elegant mechanistic study, which really highlighted the potential role of amino acid kinetics in stimulating muscle protein synthesis. So what they have here is essential amino acid concentrations over an acute time period, 320 minutes. And both conditions were 25 grams of whey protein. But the whey protein was ingested either as a bolus, which are the closed circles, or as a pulse. So regular 10 boluses, small boluses of 2.5 grams of that whey protein. So essentially, what you had now was a bolus condition that acts like typical whey protein, and then you've got a pulse condition that acts like casein protein source.
Oh shoot, that's a shame.
So essentially what you had here is the same amino acid profile, but different amino acid absorption kinetics. And what they showed is when they measured muscle protein synthesis rates, when those amino acids appeared rapidly into the bloodstream, as you would expect from a whey protein source, the response of myofibrillar protein synthesis rates during post-exercise recovery was greater compared to when those amino acids appeared in the bloodstream slowly, albeit over a longer time period. So some mechanistic evidence here that rapid amino acidemia enhances the stimulation of muscle protein synthesis after resistance exercise. If we want scientific evidence that the essential amino acids are the key amino acids in terms of driving muscle protein synthesis, we can refer to this study from Kevin Tipton back in 1999, whereby healthy young men were given either 40 grams of mixed amino acids or 40 grams of essential amino acids only, so not a combination. So the mixed amino acids had essential amino acids and non-essential, whereas this group only had essential amino acids. And as you can see, this was actually muscle protein balance being measured, but you can see that the response of muscle protein balance following exercise was greater with essential amino acids compared to a mixed amino acid source, demonstrating that the essential amino acids are the key amino acids in terms of stimulating muscle protein synthesis rates. And then finally, if we look at mechanistic studies that highlight the role of leucine, so it's fairly well established that the leucine content of a protein source is important in terms of stimulating muscle protein synthesis, given that leucine not only acts as a substrate for muscle protein synthesis, but it also acts as a trigger to stimulating the protein synthetic machinery that drives muscle protein synthesis. And here we have a study by Phil Atherton, and unfortunately, sorry, the x-axis hasn't come up correctly here, but these are the different essential amino acids, and here we have leucine. And as you can see, this was a in vitro study, when you bathe the cells with the leucine, you can see this increase in mTOR phosphorylation, which clearly shows the role of leucine in terms of triggering the muscle protein synthetic machinery. But when you put these two factors together, I think a really nice analogy to use is this analogy of the Lego in terms of the brick wall analogy using Lego, given that all of us as children probably had access to Lego or at least are aware of Lego. So here we've got a picture that essentially shows the importance of having a full essential amino acid profile and a high leucine content for stimulating muscle protein synthesis. So here we've got our brick wall, which is made up of a number of different colored bricks or Lego bricks that denote the different amino acids. At any one time, we have endogenous amino acid pool, which consists of both non-essential amino acids and essential amino acids. But if our goal is to build this brick wall, i.e. our muscle protein, at the building site, we need a plentiful supply of essential amino acids, as shown in that previous study from Kevin Tipton. We can think of the Lego person as the mTOR pathway. We can think of a cup of coffee as the red leucine brick that stimulates the Lego man or Lego lady to build the muscle protein. And if a plentiful supply of essential amino acids are available, then that brick wall will be built readily and efficiently. However, if there's not a plentiful supply of essential amino acids, and even if there's plenty of leucine, whilst this Lego person will be stimulated to build the brick wall, if they don't have the substrate, if they don't have all of the different essential amino acid bricks, this brick wall won't be built effectively and or efficiently. So let's have a look at the essential amino acid and leucine content of different plant and animal-based protein sources. So first of all, on the Y, we've got the essential amino acid content. Again, green is the plant, red and white are the animal proteins. And again, generally speaking, we can see that the essential amino acid content of the animal-based protein sources exceeds that of the plant-based protein sources. And if we actually take a human muscle itself and we actually mash up the muscle tissue, liberate the amino acids and look at the amino acid profile of our skeletal muscle, and this was a quadricep muscle, then the actual essential amino acid profile of the dairy proteins in particular are very representative of the human muscle itself. But what's also interesting is that the plant proteins are deficient in at least one of the essential amino acids, at least according to the FAO estimates of sufficiency at the individual essential amino acid level. And as you can see, the plant proteins are typically deficient in one of two essential amino acids, namely lysine or methionine, but typically not in both. Here we see the leucine content of different animal and plant-based protein sources. And again, generally speaking, if we look at whey protein, has a really high leucine content, fairly high with the milk proteins, and generally speaking, lower in the plant-based proteins, although there are some anomalies to this. So what about, we've talked about the different key factors that determine the anabolic potential, or at least in theory, the anabolic potential of a protein source, whether it's a plant or an animal-based protein source. But what about comparing the capacity of animal proteins versus plant-based proteins when matched for dose, and whether consumed in food or supplement form in terms of stimulating muscle protein synthesis? What do the scientific studies demonstrate in this regard? So to my knowledge, the first study that really directly compared a plant versus animal-based protein source in terms of stimulating muscle protein synthesis rates after exercise in trained young individuals, in this case it was trained young men, was performed by Sarah Wilkinson during her time, again at Stu Phillips' lab in McMaster, Canada. So they measured muscle protein synthesis rates during the acute recovery period from a single bout of resistance exercise. So the acute recovery period being around 4 hours of recovery, after ingesting either 18 grams of skimmed milk or 18 grams of soy milk. And what they showed was that during this exercise recovery period, the response of muscle protein synthesis was greater with skim milk compared to the soy milk. And interestingly, these acute metabolic data nicely translated to a more chronic training study, again performed by the same group. So these are data from Joe Hartman, same group, this time expressing a change in lean body mass over a 16-week period of resistance exercise training with 3 different groups, either a training group, whereby carbohydrate was ingested immediately after each training session. So they did 3 training sessions per week over a 16-week period. Training was either another group that followed training with soy protein, or another group that followed each training session by milk protein. And whilst there were plenty of individual variation in terms of the change in lean body mass, overall, the mean values in terms of changes in lean body mass were greater when milk was ingested after training compared to the soy or the carbohydrate control condition. Now, the next logical step in terms of this kind of area of determining the impact of different protein sources on muscle protein synthesis was to determine which component of the milk protein was particularly anabolic, at least in terms of stimulating muscle protein synthesis rates. So in this follow-up study, Jason Tang looked at the 2 protein fractions of dairy protein, namely whey protein, which makes up 20% of dairy protein, and casein protein, which makes up 80% of dairy protein, and compared muscle protein synthesis rate to soy protein. So each condition had 20 grams of protein ingested following an acute bout of resistance exercise, again in young men. And what you can see clearly is that it was the whey protein that was responsible for stimulating muscle protein synthesis rates over the soy when it came to the previous study with the milk protein. And again, these acute data did extrapolate fairly nicely to a longer term study in which greater improvements in lean mass were observed when whey was ingested combined with training compared to soy or a carbohydrate control. Now, those previous studies very much looked at the dairy proteins, but what about the comparison of soy protein with, say, a different animal protein, and in this example, beef protein? Now, these are data from Stu Phillips' lab. They're not actually published, but they are included in one of Stu's review articles, and I did ask for permission to present these data. What they have here in this study was actually middle-aged individuals measuring muscle protein synthesis rates under both resting and post-exercise conditions. So they performed quite an elegant protocol whereby one leg was the exercise leg and one leg was the rested leg. So this kind of unilateral resistance exercise model. And that enables you to essentially get much more information from that study. You're able to determine or compare between different protein sources under resting and also post-exercise conditions. And what you see in this study was that the beef protein stimulated muscle protein synthesis rates to a greater extent compared to soy under both resting and post-exercise conditions. And that was essentially likely due to 2 factors, namely the more favorable essential amino acid profile of the beef and also the greater leucine content of the beef over the soy. Not only have studies compared back to soy, there's also been a recent study. This was a study from Luc van Loon's lab that compared an animal-based protein back to wheat. So in this case, it was micellar casein compared to wheat hydrolysate. This was in older adults under resting conditions. And again, as you can see, there's a greater response of protein synthesis with the casein compared to the wheat. So when you take all of these data together, based on the existing available evidence, the general consensus at the time was that animal-based protein sources are more potent in terms of stimulating muscle protein synthesis when compared on a dose-matched basis compared to plant-based protein sources. However, I think that the caveat to that point is that point is very much limited to the comparison back to primarily 2 plant-based protein sources, namely soy protein or wheat protein. There are many other plant-based protein sources with quite favorable essential amino acid profiles and leucine content. And I think that there's very much a lot of interest in looking at these alternative, or at least these different plant-based proteins in future studies. And there are a couple of recent studies that I haven't included that have gone ahead and looked at these different plant-based protein sources. But most of those previous studies very much looked at isolated proteins. But a question, of course, we always get asked is, what about supplement forms versus food forms of protein? Is there a difference? And this slide is taken from a whole host of different studies that on the left-hand side essentially had isolated proteins, and on the right-hand side, more food forms of protein source. And essentially what you see is that the change in muscle protein synthesis doesn't appear to be impacted from whether the protein is supplied in a supplement form compared to a food form. And actually, really where this area is moving is to try and understand foods, protein foods, or commonly consumed protein-rich foods, given that we eat food, we don't eat supplements per se. And of course, when you look at food forms of protein, it's a much more complex interaction between the protein and the other nutrients, whether they're macronutrients or micronutrients. And this is really shown in these two studies here. This was a study from Tabitha Elliott during her time at Galveston with Kevin Tipton. And this was a study that compared milk, either provided in whole milk form or in skimmed milk form. And they measured the utilization of the amino acids and essentially showed that with whole milk, i.e. with the combination of fat and protein, that there was a better utilization or a greater utilization of the amino acids compared to when that milk protein was in a skimmed form, or whether again in a skimmed form, but energy matched. So there seemed to be something going on here in terms of the interaction between fat and protein, at least in terms of the utilization of amino acids. And this has actually recently been supported by a study, again, a really neat study with egg protein, which essentially did the same comparison. So there was one group that received egg white only, so the yolk removed, so the fat content of the egg being removed, versus the whole egg condition, versus a fasted scenario. And the response of muscle protein synthesis this time directly measured was greater with the whole egg, i.e. the combination of protein and fat compared to the egg white. So there does seem to be at least preliminary evidence that there's this interaction between protein and other macronutrients. So where next for this area? Of course, there are many vegetarian athletes, high-profile vegetarian athletes, such as Hector Bellerin here, and I know other high-profile tennis players, Serena Williams and Novak Djokovic are two examples that clearly do very well on a vegetarian diet, on a plant-based diet. And of course, this is a very much a key question, particularly when we think about sustainability, given that these are fairly crude data, but the point being is that generally speaking, from an environmental footprint or carbon footprint perspective, animal proteins are inferior compared to plant-based proteins, particularly when we talk about beef. Clearly, there are a number of factors that determine or can impact the carbon footprint in terms of the processing and the food chain. However, generally speaking, the carbon footprint is going to be higher in these animal proteins compared to the plant-based proteins. So the question is, what can we do in terms of plant-based proteins to maximize muscle protein synthesis rates if we are going to move towards a more plant-based protein diet? And of course, the first and most logical step is to perhaps recommend a higher protein dose on a per serving basis. We and others have previously profiled or characterized the dose response relationship between muscle protein synthesis rates and ingested protein sources. So this was our study that looked at the dose response relationship between muscle protein synthesis and whey protein under resting and post-exercise conditions. And essentially we saw a dose-dependent increase up to 20 grams of whey protein, and then no further stimulation of muscle protein synthesis when we increased the dose from 20 to 40 grams of whey protein. And this was evident under both resting and post-exercise conditions, at least in this group of resistance-trained young men who were performing lower limb exercise. So what's the representative per meal dose of protein for various different plant-based and animal-based protein sources if we base this theoretically on what we consider to be the optimal leucine content of that protein source? So what you have here in the black font are the number of grams of that different animal-based or plant-based protein source that, in theory, based on the leucine content of these different animal and plant-based protein sources, will stimulate a maximal response of muscle protein synthesis. But sorry, that's where the blocks are, where the bars are. But what you have in black font is the amount of that actual food that would be necessary to ingest to meet this overall protein content of the to determine the relevant level of leucine. And what you see is around 27 grams of whey protein. If we think that typically whey protein may consist of around 90% whey with the addition of carbohydrate, perhaps fat, etc. But on the other end of the spectrum, here we've got potato protein, which in theory, based on leucine content, would relate to around 2,891 grams of potatoes, which equates to around 13 pretty large potatoes. So from a practical perspective, of course, trying to optimize muscle protein synthesis rates with plant-based proteins, at least potato in this scenario, would be perhaps not considered practical. So another strategy is to look at fortifying or enriching the leucine content of the different plant-based protein sources. And this idea or this notion has been examined in the next two slides. The first one being a rodent study from Lane Norton, in which they compared whey protein with wheat protein, and then the wheat protein condition that was enriched with additional leucine so that the leucine content matched the leucine content of the whey protein. And what you can see is, as expected, there was an inferior response of protein synthesis when the dose of whey and wheat were matched. However, when that wheat was enriched with additional leucine, there was essentially a rescuing of muscle protein synthesis rates, suggesting that by enriching a plant-based protein source with additional leucine, it may be possible to essentially rescue the response of muscle protein synthesis. Now, this observation hasn't necessarily always been shown, at least in this recent human study, very similar study design, whereby in this situation, soy was enriched with additional leucine. Now, this was a study in humans, Tyler Churchwood-Venny, in a context of concurrent exercise. There are a number of different variables that could be playing a role here, but in this situation, essentially there wasn't any rescuing of the protein synthetic response. Another strategy would be protein blends. And in terms of blending proteins, this could be blending animal-based proteins with plant-based proteins. So in this situation, soy protein was combined with whey and casein. And when that was combined in a blend, there was a similar response of muscle protein synthesis compared to the whey protein condition alone. The other consideration is to simply look at the essential amino acid and leucine content of the other plant-based protein sources and look at which other plant-based protein sources based on essential amino acid content, for example, may be promising. And the other point to make, and perhaps the most logical starting point, is to think about our complementary plant-based protein sources. So here, as previously mentioned, what you have is the lysine content on the left slide and the methionine content of different plant-based proteins and animal-based proteins. The line, the dotted line, determines whether is the sufficient level for each of those individual amino acids. So below the line is seen as deficient, and above the line is seen as more than sufficient. As you can see, generally speaking, if we take pea protein, for example, pea protein has a sufficient level of lysine according to FAO, but is deficient in methionine content. If we then pair that up with corn, corn has sufficient methionine levels, but is fairly low in terms of lysine content. So we would call these complementary plant-based protein sources. And the same is true when you combine soy and hemp and potato and brown rice. And there are a number of other different combinations. And as you can see, when you put these combinations into place within various recipes, and these are recipes taken from Anita Bean's recipe book with vegetarian diets, you can see that, of course, you can make some really tasty looking dishes. So just as a final slide, of course, in terms of looking at alternative protein sources, and at the risk of opening a can of worms, I suppose, what about other types of insect proteins? There have been a lot of interest in this topic. This was a recent review, again, from Tyler Churchward-Venny, which essentially looked at the leucine content of different species of insects. And as you can see, it seemed that the crickets, the termites, and also the beetles came out quite favorably in terms of leucine content. And of course, there are a number of more social issues in terms of the acceptability of crickets as a food source. But I think that this could be an area that receives some scientific attention. So the take-home messages from this presentation, first and foremost, hopefully you didn't need convincing that skeletal muscle health is crucial across the lifespan, whether that be for performance or athlete performance or for more health reasons. Hopefully I walked through 3 key factors that appear to dictate the anabolic potential of a protein source, namely the digestibility and the amino acid kinetics, the essential amino acid profile and content, and also the leucine content of that protein source. Based on current scientific evidence, on a gram for gram basis, animal proteins, and in particular the dairy proteins, have been shown to be more potent than plant-based proteins, at least when looking at soy protein or wheat protein in terms of stimulating muscle protein synthesis. But as mentioned, the caveat to this statement is that a number of different plant-based proteins still haven't been studied. They are doing so now. I know Luke Van Loon and Stuart Phipps' lab and Ben Wall's lab are doing a great job of expanding the knowledge in this area. Several strategies exist to increase the anabolic potential of a plant-based protein source, namely enriching with leucine, increasing the dose of that plant-based protein source. So these are factors that need consideration. What about the co-ingestion of other nutrients alongside the protein? And finally, when we make these recommendations around protein for muscle, perhaps taking a more holistic approach by not only considering muscle protein synthesis or muscle health, I think it's important that we consider sustainability, appetite, and other nutrients of concern. I think importantly, this applies not only to the general population and a health scenario, but also in sports nutrition as well, whereby we've also got a role in terms of not only focusing on performance, but also in terms of sustainability. So hopefully, if you did watch the Game Changers documentary, hopefully you'll perhaps take on board that there were lots of good things in that documentary, but also perhaps some of it was sensationalized, and hopefully some of the data presented here will give you some information there. And I would recommend Asker Ukendrup put together a really nice balanced overview of summary of this documentary. I'd really recommend going to read that account. So with that, thank you very much for having me at the University of Bedford. Thank you very much to Mike and Newell for the invite, and I very much look forward to any questions that may be posed. And thank you very much for listening.
All right, thank you very much, Ollie, for that. Let me just get your. Could I get you to just stop sharing your screen? Is that all right?
Yeah, sure.
Perfect. Now, for those who have joined us on YouTube today, thank you very much for watching. Again, I can't stress this enough, but if you have any questions for Dr. Witard, then please do chirp up. I can see we've got sort of 14 or so people in the Zoom call with us today as well, so thank you very much for joining us, and hope you all enjoyed the talk as much as I did. I think perhaps if I kick off with a question, and then we can kind of politely go from there. So I thought the nice balance between this idea of like sustainability along with like the amino acid balance with this total dose that's available and how that can fluctuate depending on the sources, throwing in the environmental issues as well. So this is maybe just an open question, and just to build on what you've already kind of brought in, is that if you were to kind of design your own custom protein source, so no holds barred, like wherever you want to start, what does that protein source look like, and what kind of characteristics would it have?
Yeah, good question, Mike. I think my instinct is to refer back to the Lego picture again, in terms of that providing a full range of those essential amino acids. I think that's really key point, that without those full substrates, it's not possible to completely build that muscle protein. Of course, leucine does seem to be a player, but it may not be necessarily as important as perhaps once we considered, in terms of there may be kind of a dose response relationship in that sense. If we then go more to a sustainability side of things, I think that the way I see things is, this is on a bigger scale rather than just looking at product development, but is in the context of diet, involving mathematical modeling, for example, to try and work out, okay, if you're going to take this animal protein out of the diet and replace it with this plant-based protein, what impact are you going to have on, say, another nutrient of concern, whether that be iron or whether that be iodine or whatever that may be. Then how can we use mathematical modeling to try and combine all of these different aspects of recommendations, not only to maximize muscle protein synthesis, but also tick the box, that's a terrible expression, but to be sustainable and make sure we're covering all bases. And I suppose that's kind of that holistic approach. And it's not only muscle, it's beyond muscle. We look at appetite regulation with protein nutrition as well. So I think it's a great question.
Excellent, thank you, Ollie. That was nice. And yeah, that like taking it the next step and taking the human out of that picture, and maybe just looking at it way more sort of from a standoff perspective to try and get all of the different variables kind of balanced out, which would be really nice. Julia, who is a senior lecturer within our department, has got a question for you. Julia, over to you.
Hi, Oliver. Thank you very much for your talk. So I was interested in how you were saying that protein was really important across the lifespan for muscle growth and maintenance. So with children and adolescents, obviously they're growing, and they've got their distinct hormonal and metabolic profiles, which are different from adults. But I noticed most of the research you presented was in adults. So how well do you think that research might translate to children and adolescents? And do you think that would be important, considering that this is a population that are most concerned about our planet and sustainability at the moment, and might be more inclined to follow a plant-based diet? So if they start doing that, is there sort of research in that population evolving?
Yeah, thanks, Julia. Really good question and really topical question. There are a number of studies that are coming out in terms of protein recommendations for children and adolescents of both boys and girls. Dan Moore in Canada, in Toronto, University of Toronto, is really driving that field at the moment. So I'd recommend referring to some of Dan Moore's work. You kind of nailed it in terms of the reason why studies in children haven't been conducted, at least with muscle protein synthesis is because, of course, you can't take muscle biopsies from children for ethical reasons. So we don't know what's happening at the muscle level, but what we can do is measure. We can still take blood samples. Yeah. We can still infuse. We can still use tracers. So we can get measurements of whole body protein metabolism. So in terms of extrapolating the findings from young adults or older adults to children, you've already got that challenge that you're assuming that changes in whole body relate to muscle, which doesn't always necessarily marry up. However, if you do look at some of the work that Dan's done, looking at the dosing response, looking at, primarily dosing of protein and pattern of protein intake throughout the day, again, it does point towards higher protein intakes in children, particularly in adolescents, in that growth spurt, compared to, say, young adults. Generally, we have moved away from giving recommendations on a absolute basis and expressing them on a relative basis. So in young adults, it's generally accepted around 0.25 to 0.3 grams per kilogram body mass per meal. Whereas for an older adult, that's closer to 0.4 grams. Whereas for a younger adult, it appears that, again, that's going to be higher than the younger adult. So to summarize, there's not as much out there in children and adolescents. It is becoming more, there is more and more information becoming available, and it's pointing towards that a higher protein intake is important. Your point's a really valid one in terms of, this children being the future, and they're such an important population group in terms of choices moving forward. So I would like to see more studies done with the comparison of different plant or animal-based protein sources in children. I think that would be a logical next step for sure.
Okay, thanks. I was also really interested to see the Alliance on Potato research or something like that, which funded some of your research. Quite specific. I've never heard of it.
No, there's a number of different funding sources out there.
Yeah, no, you've got to find them, haven't you?
Yeah, that's the trickiest part.
Yeah, exactly.
I mean, you're
all right. Thank you.
Thanks, Julia.
Thank you, Julia. Yeah, sorry, you cut out a little bit there, but you're recommending 13 potatoes per dose of protein here. Do you not have a conflict of interest?
Well, no. As in, the 13 was essentially to really highlight it's not really a practical source. I'm just pulling your leg, to be honest with you. So we have a question from our YouTube viewers. So we've got Paul Morgan, who I'm going to assume is the research fellow Paul Morgan from University of Exeter. He said, "Very nice talk, Ollie. What are your thoughts, if any, on the potential of for hydrolysis in plant-based proteins to maximize the muscle anabolic response?" Yeah, great question, Paul. I think that looking at this kind of treating or changing the profile of the different protein sources is really interesting. In terms of plant proteins, I think that's a really tricky question, given that I don't think there's that much out there in terms of hydrolyzing those protein sources. What there is plenty of evidence out there in terms of dairy proteins. And I think you guys have just recently published a really nice paper looking at kind of the peptide, the role of peptides in terms of their role in stimulating muscle protein synthesis rates. In terms of your question, I'll be honest, it's difficult to really nail down whether there is something there in terms of within plant proteins. We'd certainly like to see those studies performed. It makes sense that if we change the form, then perhaps we can have a different response of protein synthesis. But to my knowledge, at least in plant proteins, that work isn't available yet. whereas of course it is in dairy proteins. I've definitely skirted around your question, Paul, so apologies, but in short, I don't really know.
Thank you for that, Ollie. The YouTube folk are about 15-20 seconds behind us, so he'll get that in about 15 seconds time. So
he won't be happy with that.
He's I'll keep an eye out on the chat on the YouTube chat in case anything. I just thought another question, and I think I'll start sort of wrapping it up because we are sort of over the 4 o'clock mark. And if anybody does have any questions, please do. Now is your moment type thing. But I think the potato example, I was kind of pulling your leg a little bit about, but I think it really highlights the kind of the food approach to protein consumption, because whilst in these studies we're trying to isolate different components of the different mechanisms of action by having elevated specific amino acids and such, like we're isolating that from the realities of consuming foods. And again, the potato example is a nice example of saying how inappropriate it is for maximizing muscle protein synthesis, but at the same time, does it represent an opportunity for the co-ingestion to meet other nutritional needs? So we're thinking a little bit along the lines of, the co-ingestion around carbohydrate ingestion and protein, which we know elevates glycogen resynthesis, but it doesn't necessarily sort of have an additive benefit of muscle protein synthesis. So with that in mind, I say, with that kind of sort of general area, where do you think there is opportunities for co-ingestion of maximizing muscle protein synthesis, but also meeting other nutritional needs? Do you have any thoughts around that?
Yeah, that's quite a tricky question to answer succinctly, at least. I agree with you in terms of the future, looking at food forms for sure, and understanding these interactions between the different nutrients, because as you say, we eat food, so understand that interaction. That's definitely almost one part of it, and understanding those nutrient to nutrient interactions is really complex. They're complex mechanistic studies that we've got a few good ones out there, but certainly more and more interactions, not only in terms of nutrient interactions, but what about the effect of say chewing down that food form, or how that's delivered, essentially, whether it's in a solid or liquid form. These are all interactions. So that's one aspect of that. The other aspect is then how does that impact other aspects beyond muscle? And I think, again, that's where we need to sort of think about this modeling approach, have more multi or interdisciplinary studies where we don't just look at one outcome, i.e. muscle protein synthesis. We look at the bigger picture of appetite, or whether it's insulin resistance or glycogen resynthesis in a sports nutrition perspective. I think the key there is to try and design studies that answer a question, a practical question, rather than maybe, you working on your field of carbohydrate, me working on my field of protein, and not talking to one another. It's much better if we can design these more like holistic kind of studies that answer a practical question. Again, going back to Julia's point though, finding the funding to do that in sports nutrition at least is really tough. So, but yeah, I like your thinking, Mike, in terms of answering the bigger question and seeing what impact it has on different aspects of performance, for example.
Perfect. Thank you for that. A tricky question, I know, so I won't push you anymore on that one. We do have another question, and it's probably the final question, I think I would say at this point. And it's from a University of Stirling alumni, Andy Shaw. So we'll throw it out there. So first question, and is a very relevant question: how many beetles has Ollie eaten?
Yeah, I have been fortunate enough to try some of the insect protein, and was pleasantly surprised as to how palatable it was. And I do know, again, from talking to Luke Van Loon in Maastricht. Maastricht University is based at the med school, based within the hospital. They have insect protein in their in the hospital, in terms of in the available to the public, and I think it's fairly popular. So whilst I get Andy's point, I think there are ways with food processing that it can become more acceptable. That surprises me to say that, but I think it could be okay.
He does go on to ask a slightly more grown-up question in that. So he says, do you know what, if any, mitigations are made by top-end veggie athletes to compensate leucine levels? And is there any evidence that veggie athletes have less muscle mass on average, or lower score on any physical performance variable? So I think it's what mitigations are in place for veggie athletes? Do they compensate for their leucine levels? And is there any evidence that they have different physiological compositions or different performances, basically?
Yeah, several really good points. From an applied perspective, unfortunately, I haven't been in the applied field for a while at least, so wouldn't necessarily have that information, but would be keen to hear and find out. So if anyone is listening that has some views on that, that would be really interesting. In terms of the question regarding average levels, again, I'd point towards some recent papers that have profiled typical dietary protein intakes in athletes. A recent paper, again, from Luke's lab that looked at this. Also a paper by Michael Teeland, although that wasn't in athletes so much. So there are papers out there in terms of profiling typical protein intakes. Of course, you don't hear about these papers as much. You hear about the extreme scenarios of athlete X who has this amount of protein. We can take the Game of Thrones as one example, where that came out and was published, and we actually wrote a conversation article of what that might look like in terms of the mountains protein intake. So yeah, again, I appreciate I've skirted around the answer, but essentially, in terms of the applied aspect, unfortunately, I'm not aware of that kind of area, but I'm sure there's many people that are much better placed. In terms of average intakes, there are some good papers out there that you can refer to. I'm trying to think of the name of the first author, but her name escapes me. She's based in Canada, and she came over to Maastricht for a year. I can put it in the chat or something if her name comes to my mind.
That's all right. You've been on for the last hour and 15 minutes now, so you can be forgiven. So, all right, we don't have any other questions from the YouTube chat, and I don't think there's any questions from internally either. So I think it would be a good opportunity to sort of politely say thank you very much for your time.
Thanks, Bob.
Yeah, and everybody on YouTube, thank you for watching, and the video will be available afterwards for sharing more widely if you think it would be useful. But thank you very much.